Okay, good morning, everybody. I see there's still people registering in, but I want to try and keep this on time. Thank you very much. My name is Russell Poole. I'm the section manager responsible for the management of the Marine Biodiversity Scheme and the Marine Knowledge Scheme, and I'm going to try and chair this webinar remotely. We've had a huge response. I don't know how many will actually register at the meeting, but there was over 160, which is possibly more than double any of the other webinars we've had in the last 3 or 4 years. So that's great. Thank you very much. Okay, so a quick overview of the European Monetary Fund for Aquaculture and Fisheries, the EMFAF. It's a scheme, an operational programme funded by the EU and matched by the Irish government to run from 2021 to 2027. And this webinar is the first of our MFAF ones, and we will endeavour to have 2 a year from now on. So the overall programme is worth about 142 million EU contribution to the Irish economy, and that's matched with 116 million by the Irish government, of which 16 million is for the biodiversity and marine knowledge schemes, and about 44 million plus core 20 million funds the DCF, the data collection framework for fisheries. The Marine Biodiversity Scheme, it's a EU Priority Area 1 scheme for fostering sustainable fisheries and the restoration and conservation of aquatic biological resources. It's mainly for the protection and restoration of aquatic biodiversity and ecosystems. And under this scheme, we're combining fisheries and aquaculture. The EU lays out a series of interventions and projects which we put our projects under these headings. So the headings are reducing negative impact and contributing to positive impacts on the environment and contributing to good environmental status. The intervention 6, contributing to a good environmental status through implementation and monitoring of marine protected areas, including Natura 2000. So this essentially is projects to support our Natura, MSFD and sensitive species listings where they come in contact with fisheries and aquaculture. We have one project on control and enforcement, and I think Ollie Tully will be describing some of that in his talk. And then we have one project under data collection, analysis and promotion of marine knowledge, which is our outreach program, and the webinar falls under that. In the other scheme, Marine Knowledge Scheme, it's basically to strengthen sustainable sea and ocean management through marine knowledge promotion. It's a Priority Area 4 programme, strengthening international ocean governance and enabling seas and oceans to be safe, secure, clean and sustainably managed. And the specific objective that the knowledge scheme falls under is the intervention that contributes to strengthening sustainable sea and ocean management through the promotion of marine knowledge, maritime surveillance, or Coast Guard cooperation. And the key, there's a list of actions in the legislation, but the key ones here are data collection, access and analysis for helping us deliver national obligations and measuring changes in the environment, and improving the understanding of impacts of human activity, including climate change and the impact on marine environment. So the webinar today, we'll have talks on the Marine Knowledge Scheme, the Marine Biodiversity Scheme and the DCF. And because the Marine Knowledge Scheme and the DCF are both EU operations of strategic importance, which means that there's a focus on outreach from those projects. It's to improve connection between the projects and the general public. I'd like to welcome officers from the EU if they're attending this webinar, because we have invited members of the EU. We're recording this, and the talks will be made available afterwards. A number of people are at, for example, the Fisheries Council meeting in Brussels yesterday and the day before, and are traveling today, so they couldn't be here. So we said we would make those links available. There's a Q&A. If you have burning questions, put them in the chat, and we'll try and pick them up if we've time at the end of each talk. And if not, we'll collate them and respond to them after the webinar by email. We have a number of EMFAF videos, and we're filming another one at the moment on marine knowledge. The biodiversity, fisheries, biodiversity, aquaculture, and an intro to EMFAF. These are all available on the Marine Institute EMFAF YouTube channel. So if you search the YouTube channel for MIEMFAF, you should come up, and the links will be in the recording of my talk anyway. And our new website will be open in early 2025, and that will have full information on all the projects that are going to be running in the program. So you have the agenda today. So we'll kick off with our first talk. Our first speaker is Professor Philip McGinnity from UCC, University College Cork. And he's a biologist working principally on the areas of fish population genetics and fish habitat relationships in support of management and conservation of Atlantic salmon. His talk is about a project that he's running under Priority 1, the Marine Biodiversity Scheme. It's establishing a genetic baseline to assess gene flow from salmon farms into wild Atlantic salmon populations. So Phil, if you want to, I'll unshare and you can share your screen. Thank you.
Thanks very much, Russell. How does that come up okay?
Yeah.
Oh, that's great. Okay, thanks very much for the invite, Russell, to speak to the webinar this morning. This project's about, as I say on the title here, establishing a genetic baseline that would be useful for assessing gene flow from salmon farms into the wild Atlantic salmon populations in the country. And let me see, is it going to run forward? Okay. So the motivation, I suppose the background is there have been a number of international reports recently from Norway, from Scotland, from Iceland and Canada, you know, the main salmon producing areas in the North Atlantic. And it was just felt that it would be important from an Irish perspective to carry out a similar type program. Particularly, I suppose, in fulfilling Ireland's reporting obligations to NASCO and other international bodies. Some of the reports, you know, they're fairly concerning, particularly, say, in Norway, where there are integration rates. When we talk about integration, we're talking about where you would have an escape, you would have hybridization between the farm fish and the wild fish. That would be in the first generation. And then into the second generation, what we call integration, where that genetic material, that flow from the farm into the wild is retained into second and subsequent generations. So there has been some concern about the levels of integration that they're seeing in Norway in particular and in Scotland. And there have been a number of reports also about the changes in the biology of the fish as a consequence, changes in age, schedule of maturity, smoltification, sex ratios, and so on. And Ireland's not immune from this, as recent reports in the media. There was an escape back in the summer from in Killary Harbour, and there was a lot of concerns about that. So, Joe, somehow it would be good to establish a baseline to be able to compare populations before and subsequent to such events. From a biodiversity point of view, salmon are organising this kind of a portfolio of diversity, this interspecific diversity. Across Europe, where there's some 2,500 populations, each of those are genetically distinct entities. They're unique. And they are incredibly important for the resilience and productivity of the species into the future, particularly in an era of climate change, that would be protected. Integration from the farm, gene flow from the farms would be a threat to that diversity. And you see here, if you take here on the extreme left-hand side, somewhere like the Moy, there's at least 5 distinct evolutionary significant units within, say, something the Moy. Within Ireland, whatever, 200 populations or 140 populations, 150 populations, possibly up to 200 populations that are distinct. And then as you bring that into the European context, then you're into 2,500, 3,000 distinct populations. So we're kind of trying to think about how to protect that. So the question is, Joe, so what? Joe, what's the issue? What's the problem? And historically, when people would have thought about this problem originally, or the potential impact, they would have thought maybe when you take 2 distinct lines, say here, the farm or the wild line and a farm line, and you cross those, that you would get this heterosis or this hybrid vigor. Sometimes when we think about wheat and other both plant and domesticated livestock and things like dogs or horses, whatever, when you cross 2 outbred lines, you usually get vigor, or quite often you get vigor. However, it's also the potential for what they call outbreeding depression. So when you get cross 2 distinct lines, you actually get an intermediate value in terms of fitness or performance of those 2 lines. So as you move into subsequent generations, into an intergress population, where you go into F2 and F3 and subsequent generations, you get what they call this intrinsic outbreeding depression, where you get a breakdown of gene complexes and so on, and you can then have very, very significant and substantial reductions in fitness. And for many years, we've been working on with colleagues in UCC and the Marine Institute and Inland Fisheries Ireland and so on, have been working on this particular question using a common garden experimental approach. That approach facilitates you to isolate environmental variation and then look at the genetic differences. And when we look at those differences, and these are experiments we've run over many years, we see very substantial differences in the fitnesses of the progeny of these fish in the wild. So it's a big thing to be able to run those experiments, but particularly to run them in the wild. And we see in this particular box here, if you take the wild, and let's assume that the wild fitness is 1, lifetime fitness is 1, what is the relative fitness to the farm in those wild environments? It's only about 2%. So it's a very significant and substantial reduction in fitness. However, hybridized populations, we might see this bar graph here, those hybridized or first generation, or where you would have 50% of the genes are farm, 50% of the genes are wild. Progeny with that kind of profile, we see intermediate fitness. So it's actually this extrinsic breeding effect that we see, not heterosis, but we actually see a substantial reduction in the fitness of hybridized fish individuals and hybridized populations. We continue to do those experiments and those studies where we're trying to look at the mechanisms that underpin those differences, particularly in the context of climate change. So we project over the next century or so substantial increases in temperature, changes in flow regimes in terms of droughts and floods and so on. And we see that a potential for interaction between gene flow from the farms and changes in the environment that actually exacerbate the effect. So you get a bigger impact, bigger differences in fitness as a consequence of climate change. So those are studies that are ongoing in the background. So as part of this, then through the MFAF program, we have a new project, or ongoing project now. We're into our 2nd year. It's a joint project between University College Cork, Inland Fisheries Ireland and Teagasc. And it has a number of tasks within it: the design of the project, the collection of the samples, the genetics, the profiling of the wild populations, and then the interpretation of the genetic profiles and the reporting of those those levels of genetic integration. So the sampling strategy is fairly comprehensive. There's a number of different elements. One is we were very anxious to have a national coverage. Secondly, we were interested, we actually have a baseline that was prepared or developed some 20 years ago that we wanted to repeat some of the rivers and locations that we sampled in that program. Also, Ireland has based on its history and its post-glacial history, there are potentially a number of phylogeographic groups, maybe 2 different groups, 3 different groups that may have impact on our capacity to assign fish to different rivers and so on. Many of the rivers, the larger systems, say for example the Moy, have multiple populations, so we want to sample for that. There are big rivers and there are small rivers. You may imagine that a small river with a small population is more vulnerable than a big river. We wanted to focus then also on salmon producing areas, principally on the west coast, north and south, and then to target or to sample the hatchery, those hatcheries or the hatchery producing areas. So somewhere along the line, we officially we believe there's 144 salmon rivers. We would sample 200 sites, and that would approximate about 10,000 samples overall during the project. So the project has been progressing very well. IFI have 2 teams in the field last year and this year. They sampled about 90 sites last year, 56 sites this year, of which there's about 44 had salmon present, because some of them were sea trout rivers and small rivers. We were just interested in having that broad, comprehensive view. So that has gone extraordinarily well. The sampling program is well advanced. And then in terms of the genetics, what we're deploying is a set of diagnostic markers. They're SNP markers. These are the single nucleotide polymorphisms. They're diagnostic to discriminate between the farmed and the wild. Some of the markers were developed in previous projects that were supported by SFI. But we believe, a very powerful set. But we had to optimize those. So there's a number of different steps. The first step, and that was the SNP marker validation and optimization. We do that in-house in UCC. Then for the large or the high throughput screening and the high potential screening, we use colleagues in Chagas in Moor Park. They run it on these very high throughput systems. Here we have the Illumina system, the MySeq system. That data then comes back to UCC, the bioinformatic data, where we then we attempt to interpret the genotypes and look at the integration levels. And then down the line, and I see a mistake here, autumn 26, but hopefully autumn 25, we will report on the various on the survey of the Irish populations. So one of the first parts of that is the validation of the markers. We believe the markers are very, very powerful. They can distinguish, and here we see here on the right-hand side, on the left-hand side here, Joe, the profiles of the farmed versus the wild, they're 2 very distinct assemblages. So we can assign with very strong confidence. Here's just a few trout down here to just show that they're quite different profiles from the marker panels that we have. And then we simulate different mixtures to look at F1s, F2s, back crosses, so that we can have an idea of the patterns that we might observe. So to finish up, what's the plan? Where are we going 2025? Well, one is we're always continually refining the wild baseline. Joe, we have this question here, we see here on the left about, Joe, the legacy of the Ice Age and the structure of the populations, where Joe, if there are 2 quite different or distinct phylogeographic groups, we want to take account of that. We want to optimize the markers as far as possible. The sampling in 2025 would probably be driven very much by what we see in 2024 and 2023. So where are the areas that might have potential for integration that we might want to look at again in a second generation, to look more widespread throughout a system, into the headwaters versus the lower reaches of those rivers. So we will kind of optimize the system in 2025, the sampling approach in 2025. And then I talked a little bit earlier about this baseline that we have from 2005 and 2006. And actually, that was it was a question about diversity and so on. If some of you might remember back to the drift net fisheries that Ireland prosecuted back in the 60s, 70s, 80s, 90s, up into the mid-2000s. At that time, Ireland was very much the bête noire of the salmon fishing communities, both angling and conservation, where it was presumed that Ireland was intercepting fish from all over Europe. However, based on the genetics and this type of technology that we're deploying in this study, it didn't turn out to be that case. Ireland was actually intercepting very few non-Irish fish from, say, UK, France, Spain, other parts of the world, Norway. What we were prosecuting or were targeting fisheries was in a mixed stock fishery. So, for example, here, let's take this broken line here as Donegal. Fish that were being captured in Donegal contained or had fish from rivers from Dublin, Wexford, Waterford, so on. So we were actually hurting ourselves more than we were internationally. But that kind of data, that empirical data, that evidence-based data was facilitated then some major policy changes and changes in the nature of the commercial fisheries. So I'm not doing all this work myself. I'm very much just front-ending it. The teams in Cork are Jimmy Coughlin, Oliver Stewart, Eileen Delan, Tom Cross, Tom Reid. In the fisheries, in inland fisheries, Mick Milland, and John Coyne and his river sampling team, Alistair Dudman and his team, and then Fiona Crispy and Chagas. So, Russell, I don't know how we're doing for time there. Thank you very much.
That's great, Phil. Thank you very much. I don't see any immediate questions online. Julie, correct me if I'm wrong. Phil, just waiting to see if a question comes up. I'm not wanting to put you on the spot. The escape in Killary that you mentioned in your first slide, is that likely to show up in your surveys? Or are the timings of your surveys right for detecting that?
Well, I think the timing is very good that we have the baseline in before any potential change or gene flow that would happen this spawning season. Although our expectation is that, Joe, a lot of those fish, from what I understand, they were non-maturing fish. I think they were this season's fish, and I think they were scheduled to mature next year. So yes, definitely. So the baseline is there. If there's any changes accordingly, we should be able to pick those up. And we have been looking at samples. IFI, our partners in IFI, have provided us with the fish that from the escape that have turned up in different rivers. So we have those profiles, and that it'd be a quite powerful and great opportunity to compare what was there before and what is there now.
Thank you very much. And I have one quick question from the chat. Do you think there'll be any policy change in the use of sterile farm salmon in the future? And I just to broaden that, I think they've already started doing something like that in things like Pacific oysters, so that they don't breed in the wild with climate change.
Yeah, it's very much, I would say it's a very real possibility, particularly now when you start to see this evidence-based, Joe, these reports from Norway, Canada, Scotland, Iceland, ourselves in the future. That would be a very, very powerful approach to preventing gene flow or escape. And I think many of the audience may be familiar with this idea of triploidization. There's a way of sterilizing the fish and so on. So yeah, that would be a useful approach.
Super. That was a question by Stephanie Linehan. Okay, I'll draw you to a close, Phil. Thank you very much.
Thanks, Russell. Thank you.
That's great. Thank you. Our next speaker, hopefully he's online, is Dr. Oliver Tully. He's a team lead in FIAS, the Fisheries Ecosystem and Advisory Services Group in the Marine Institute. And he's working on shellfish fisheries assessment and management options and effects of fisheries in coastal SACs and SPAs. His talk is this morning on spatial data on inshore fisheries, importance for marine spatial planning and fisheries management. So, Ollie, if you're there, take it away. Thank you very much.
Yeah, thanks, Russell. Can you see the screen?
We did, and now we don't.
You don't?
Now we do. Yeah, that's
okay.
It. Lovely. Thank you very much.
Okay, thanks for that, and good morning, everybody, and thanks for joining the webinar. Yeah, I'm Oliver Tully. I work in the institute here, and I'm talking about a project on spatial data for inshore fisheries. It's a short title, a simple title, with a project that has a very simple objective, really, in improving data acquisition for vessels for which we don't have very much data at this point. So what do we mean by fisheries data generally, or for data that might originate from commercial fishing vessels at sea? So what do we need to find out about vessels when they go to sea? And the data structure is simple, really, and it's at its basic level has three kind of elements. You know, where's the vessel going? What is it going to do when it gets there in terms of what gears is it going to deploy into the environment, and what is it going to catch, and in terms of the species composition and the quantities of catch that it's going to take. Now that can be variously complicated or simple depending on the type of fishery and the type of gear it is. So the vessel may go to many different locations on a given trip or a given day. They may even swap out gears and use different types of gears. And the species list could be simple, one species, or it could be multiple species. So there are variations depending on the fishery that we're talking about. But the most important thing really is to consider what is the most efficient, low cost and accurate way of acquiring these data. And in particularly, it's important to consider the least intrusive way. And by that I mean, how many questions do we need to ask the skipper to provide this data to us? And ideally, we would need to minimize that, because it's everything recording of data in a manual sense on board a fishing vessel may involve error in data recording. Can we read the skipper's writing? What happens to that hard copy data when it comes on shore? Somebody else has to digitize it. Is there error involved there? So error propagation through the whole process of data acquisition to reporting is something really to watch out for. And I guess as technology develops, can we reduce the number of questions we ask the skipper? Can we be less intrusive? And can we let him go about his business without asking him 1,000 questions every day, basically? So that's what this project is kind of about, but as applied to the coastal fleet itself, vessels under 12 meters, which are particularly data poor. So the types of data coming from fisheries then, in terms of particularly location and the fishing effort that might occur at that location, has there's a broad number of applications and needs for these types of data. Well, the first one, of course, is fisheries assessment. It's the basic data for fisheries, really, in terms of what is taken out of the sea, what amount of effort is put in to get that catch, and the location at which it occurs. So there's multiple applications in fisheries assessment, but also in fisheries management. So a lot of fisheries management involves control of catch, but there are alternative, what we might call input control mechanisms as well, which we might want to control fishing effort. So how do we control fishing effort if we can't see the boat and the boat isn't reporting fishing effort in real time? So these kind of questions are there in all fisheries. And improvement in technology and data can solve a lot of them. And of course, then the other reasons we need these data is because of the increasingly complex array of activities that are going on really in the marine environment. Obviously the offshore renewable sector will be developing in the environment over the next 5-10 years, in the Irish Sea and Celtic Sea in particular. We have government policy to expand the marine protected area network to protect biodiversity. So all of that is a marine spatial planning problem, and in the marine spatial planning process, it's important to minimize trade-off costs between these different sectors. So you can only minimize trade-off costs if you have good spatial data on where everything is. So that's a basic reason to have really good data on the coastal fishing fleet. In other fine scale adjustments, you might consider, in relation to how fishing interrelates with aquaculture installations, ORE, MPA boundaries. Fine scale spatial data could be used for fine scale adjustments to minimize trade-off costs at local level. When we already have an MPA network, of course, in the special areas of conservation and special protection areas in coastal environments, and with an ambition to expand that network, we will need evidence on as to where fisheries are operating in these SACs or MPAs, and particularly around sensitive ecological features. We have to verify that we can and that we have confidence that that these activities are not impinging on those features. And even, you know, evidence for fisheries effects on marine infrastructure, we can expect increasing amounts of infrastructure in terms of cables, various infrastructures associated with offshore renewables to be in the marine environment in the next 10 years, and we need to protect those, and fisheries also need to be protected from those in the future. Okay, so what does the Irish commercial fishing fleet look like, and what does it report at the moment? So the bar charts here show you the size profile of the Irish fleet. There's about 1,900 vessels in the fleet, and it's pretty stable, although the numbers of vessels certainly, particularly over 12, is declining somewhat. Over 18 metre has declined last year through a decommissioning program, and so, but about, you know, 3/4 of the fleet is under 12 and don't really report any useful spatial data. Vessels under 10 don't report any data other than their catch or their landings is subsequently captured in sales note information. The vessels between 10 and 12 report a logbook in hard copy, but the spatial data and the effort data in that logbook data is pretty poor. And the vessels over 12 do have a good, we have good data on that in terms of logbook data on catch and effort, and the VMS data, which is the vessel monitoring system data, which reports the vessel position every 1 to 2 hours. And, you know, that's good spatial information in that case. But if we categorize it a little bit more in terms of the spatial resolution, and I think spatial resolution is the key thing here. If we look at back to the reasons why we need this data in the first place, in terms of trade-offs with other sectors and protection of ecological features at small scale. So we can categorize them into very high spatial to high spatial to low spatial or very low spatial resolution, let's say. So this project that we're funded by through MFAF, the IVMS project, does acquire very high spatial data. So we're reporting the vessel position every 1 to 10 minutes. We have that deployed in all Irish vessels fishing for razor clams for the last 10 years. And the second kind of category there in high spatial resolution would be let's say the over 18 metre vessels, which are pinging at 1 to 2 hours, which is less resolved. But then again, most of those vessels are working offshore where the resolution doesn't probably need to be as high. It's a BIM have started actually a new participatory mapping project in the Celtic Sea, which is trying to get good spatial data through an interview process and a participatory mapping with the skipper to identify the kind of the territories of each individual vessel and stacking those territories on top of each other to provide heat maps of fishing activity. And then everything else beyond that is really low resolution spatial data, or where there is no spatial data at all. So the MFAF IVMS project basically is set up to acquire GPS data from vessels under 12 meters. And it does this through, basically we go to the market, we procure the systems from the market, we install the units on the vessels, the data that's being transmitted there in terms of the location, speed, and course of the vessel every 1 to 10 minutes. The data is transmitted over the mobile phone network through the mobile phone masts into the service provider database, and we've used many different service providers over the past 10 years. There's usually a user interface into that database where the skipper or the vessel owners or the Marine Institute or the SFPA can access and have a visual of in real time of where the vessels are. And then we download that data to an internal Marine Institute SQL database and derive our data products from that. So our purpose initially, and this project predates really MFAF and even EMFAF, and it was funded by Department of Ag, Food and Marine back as far as 2014 or so, or basically was to evaluate the feasibility of acquiring VMS data from the Irish under 12-metre fleet. There's about 1,000 vessels, active vessels in that, and it's very pragmatic. You know, what about the costs? What about depreciation, reliability, maintenance, overhead? You know, we didn't know anything about the systems that were in place at the time, and the UK and France and other countries were experimenting with different systems as well at that time. So we were also testing the market as to, you know, whether what was available. There's very few suppliers in Ireland supplying these systems. So we went through various business models, from multi-supplier frameworks, single suppliers, type approved systems that they used in the UK, for instance. Trying to integrate data across a number of different suppliers. And basically then, you know, what would we do with this data in terms of trying to develop data products and use development to support existing legislation. And as Russell was explaining, in preparation really for the EU control regulation, which will be implemented in Ireland from 2026. And the responsibility for that lies with the SFPA. This is our current system. It's a system provided by CLS. They're based in Toulouse in France. It's a solar-powered system called NEMO. I don't know why they called it NEMO, but something play on word like finding something, I guess. But so we're trying to find the vessels here. And it's a simple self-installed system with an internal solar panel, which is autonomous of vessel power, at least from the months of March to November. We've found a deficit in the solar panel doesn't maintain power over winter quite adequately. So we've more recently developed an external solar panel to attach onto it, which then makes the unit completely autonomous of vessel power, which is, from experience, and we've learned that is absolutely necessary. And that shows you we have about 100 vessels reporting at the moment. Most of them are razor clam vessels dredging in the Irish Sea. So then it gives us really good spatial data. So what can we do with that? Well, obviously we can map the entire distribution of a fishery and a fleet that is active in the fishery. And so on the top left, you'll see the distribution of the razor clam fishing activity in the North Irish Sea, from the North Dundalk Bay all the way down to Howth in Dublin. And like, we've been using these data to design our own surveys. You know, we sit our own surveys on top of this gridded data to design stratified random approach to surveying. And it also supports the SFPA in identifying which production area that the vessels are operating in. Really importantly, then, we can derive also through modelling of the spatial data coming in at very fine resolution. We can model the fishing effort, or estimate the fishing effort through a modelling process, rather. So we can identify individual operations at sea and calculate the amount of fishing effort in terms of the meters, hundreds of meters, kilometres of dredging, or the numbers of lobster or crab pots being hauled, for instance. And we've developed an R-Shiny app through a separate project, the informatics project in the Institute, which we call pings to pots. In other words, translating GPS pings to coming from the vessels to the number of pots a fleet of lobster or crab vessels might haul in a given day. So immediately that enables you to, for instance, calculate effort at sea in real time, and opens up the idea of fishing effort management also in real time. Another just an example of a use case example here, where we're showing compliance around a closed area in Dundalk Bay. So on the extreme left, we have the raw VMS pings sitting on a proposed closed area at the time in 2022-23, and in the middle plot, it's gridded VMS data underlying the proposed closed area. And then on the right, we see the heat map of fishing activity in the last 20 days. So it's like from mid-November to early December. And you can see there's a high degree of compliance with the closed area during that period. So So these small-scale closed area are legislated for through Fisheries Natura declarations by DAFM, and there's a number of them in existence throughout in bays around the coast in special areas of conservation. And you can see the importance of having really, really high spatial resolution data to support the compliance with such areas. Okay, let's twist it on its head and say, well, what's the implications if we don't have these data? And
2 minutes, Ali.
Will do. So we can't use effort management really as a fisheries management tool if we don't have it, because we will not be able to verify the amount of fishing effort at sea. The fishing footprint of the inshore fleet will not be known. We won't be able to do a trade-off with other sectors using these data. The protection of closed areas will be difficult. And we might have a kind of a sledgehammer approach really to the or a high level of precaution will be needed in for instance, providing large buffers around protected areas because we don't know exactly where the fleet is. So we need to be, in that case, we would have to take a more precautionary approach. So in summary, where have we got to? Well, we've used various systems, from many suppliers in the market over the 10 years, and we've a good idea now of cost, the fault rate, the reliability, depreciation, or otherwise, and what the market can supply. We know the main pitfalls are there. We absolutely need to be autonomous of vessel power because the vessel power itself is so unreliable. We've developed various data products and use cases in support of legislation. And we know we can get these location data, fishing effort data, unobtrusively without asking the skipper any questions. And of course, that gets us 2 of the 3 components of the fishing record. What's left, of course, is the catch data, which we still need to get manually, but we've started to think about maybe machine learning methods and camera systems where we could get catch data also in an automated kind of way from these vessels, given that many of these vessels have very simple catch profiles. So that's where we're going to go with it next. Thanks, Russell. Okay.
Super, Ollie. Thanks very much. At the moment, I don't see any questions. So I think we'll move on. Thanks a million, Ollie, for that. Very interesting. Okay, our next speaker is Dr. Catherine Jordon. She's a postdoctoral researcher in remote sensing. She previously completed a Cullen Fellowship PhD with the Marine Institute and the University of Galway on space-based observations of marine phytoplankton in Northeast Atlantic waters. So I think if you're a shellfish farmer, you should be tuning into this talk. I've seen a preliminary, it's very interesting. Okay, so Catherine's talk is Improving Ocean Monitoring with Remote Sensing, a New Approach for Irish Waters, and it's part of the Marine Knowledge Scheme. Thank you, Catherine.
Thanks, Russell. Can you let me know if you can see the slides there?
We're seeing the slide layout, but not the presentation mode.
Perfect.
Yeah, that's good. Thanks.
Perfect.
Right. Thank you.
Okay, so thanks very much for having me. And as was mentioned earlier, like these talks, they're only 15 minutes long, I suppose. So my email is on the screen there if anyone would like to contact me afterwards to talk more about my talk or any of the topics in it. So my name is Catherine Jordon. I'm a postdoctoral researcher in remote sensing in the OCS department in the Marine Institute. And today I will be, I'll be talking primarily about ocean color, which is one of the parameters you can measure from remote sensing, as this is what we've focused a lot on in the first year of the EMFAF project. So as you can all imagine, there's lots of different parameters you can measure from satellites, which I'll briefly mention, but today we'll be talking mainly about ocean color. So I'll give everybody an introduction to remote sensing for ocean waters, focusing specifically on ocean color, because I can see there's a lot of people in the room, so I'm sure there's a lot of different specialities. I'll introduce an algorithm that we have introduced to Ireland for the first time called the red band difference algorithm. And this is really useful for monitoring chlorophyll fluorescence at the ocean surface. I'll introduce ocean color satellite validation work we're doing at the Marine Institute, and then briefly introduce a new EMFAF PhD project that has just started in ocean color as well. So just a brief introduction to remote sensing for those who are not familiar with what remote sensing is. So it's the use of satellites or airborne sensors to collect data about the Earth's surface or the ocean surface without physical contact. So we collect remote sensing data using satellite data, but also using sensors on our national research vessels, which I'll introduce in a little while. So why use ocean or remote sensing for oceans? Satellites basically provide a bird's eye view of the ocean in general. So this is a really useful tool, especially for the Northeast Atlantic, which is really, really big, and it'd be very hard to sample all areas at one time without the use of remote sensing. And also remote areas, and especially with the weather that we get in Ireland, it's a very useful tool to just take one single measurement of a vast area. Using this type of data, we can create time series of multiple locations at once and look at different trends and changes in our oceans. And as I mentioned earlier, multiple parameters can be derived from satellites such as ocean color, sea surface temperature, sea level height, and phytoplankton functional types, which I'll introduce at the end as well. So how do we measure ocean color remote sensing? So measurements of ocean color are based on electromagnetic energy emitted by sunlight, which is transmitted through the atmosphere and is reflected back off the ocean surface. So depending on what's actually in the water, whether this is phytoplankton, suspended particulate matter, color dissolved organic material, different things happen to the light depending on what the light interacts with. So some things will absorb the light, some things will scatter the light. And for the ocean color measurement, we measure the light that reflects back off the ocean surface. And for those, again, who aren't familiar with what phytoplankton are and why do we monitor them, phytoplankton are tiny microscopic plant-like organisms that we generally can't see with our eyes, but we can usually see them under a microscope. But when conditions are favorable in the ocean, these phytoplankton tend to bloom, and if the bloom is big enough, we can see it from space. So we can see some blooms from space and others not necessarily. And why is it important that we monitor these? Phytoplankton are really important organisms in our ocean. They produce oxygen, they remove CO2, and they're the base of the food web, which all sounds great, and phytoplankton are brilliant for those reasons, but there are a few species that can produce harmful algal blooms by releasing toxins into the water and also removing oxygen out of the water. So for these reasons, and for the health of our oceans, it's important that we monitor phytoplankton. Now, in order to monitor phytoplankton from space, we use chlorophyll algorithms. So chlorophyll is a pigment that's found in phytoplankton. And there are different types of chlorophyll algorithms, some that are tried and tested, and then some that are new. So for Ireland in particular, the red band difference algorithm, which is the image furthest on the right, is what we have introduced through my PhD and then through the EMFAF project. We have developed a product using this algorithm. So the difference with some of these algorithms, you'll see the first two images on the left and the middle of the screen. These were determined using multiple satellite retrievals over Irish waters, but using the OC5 algorithm, which is basically looking at the ratio of the difference of the blue and the green. So the more green in the water, the more chlorophyll in the water, basically. And this algorithm has been tried and tested in open waters, and it's a really useful algorithm. But the closer you get to the coast, the more complicated it gets, because there's a lot of interference of absorption from color dissolved organic material, which is basically runoff from land, like clay and silty material and leaves and different things that run off from the land. And this can actually interfere with this signal. So it can actually overestimate chlorophyll in coastal waters especially. So for this reason, we tried and tested a new algorithm called the red band difference algorithm, which basically we look at chlorophyll fluorescence in the red part of the visible spectrum. And in this part of the visible spectrum, there's no interference with color dissolved organic material or sediments. So generally what you're seeing at the ocean surface is phytoplankton. So you can be a lot more confident that it's not an overestimation of phytoplankton in those areas. So you can actually see the third image on the screen is a lot sharper than the other two images because there may be an overestimation of chlorophyll, and they were actually taken on the same day. So this algorithm, as I mentioned, is called the red band difference algorithm. We've published a paper and it's linked in this slide here, if anyone would like to read about it. As I mentioned, we trialed it in Irish waters during my PhD, and basically what we did was we used it to track and monitor the movement and the development of a bloom south of Cork in June 2019. So you can see the first image on the left there. You can see where the bloom just starts to appear at the ocean surface, and we were able to see it develop in size and concentration over the month, and then when it finally peaked offshore just at the end of June before it dissipated. But because this was the first time we tried this out in Irish waters, we had to validate this algorithm to make sure that it wasn't an anomaly we were looking at from satellites, and that it actually was a phytoplankton bloom. And just to mention, NOAA in America use this algorithm routinely to monitor HABs. So it's been tried and tested over in America, and that's where we got it from. So in order to validate it, we did a few different approaches. We ran a particle tracking model by taking a shapefile from a satellite imagery and dropping 50,000 particles in the area that we first seen the bloom appear at the surface. And we let this particle tracking model run for 27 days based on the same kind of time that we would have seen over the satellite imagery, just to see where the bloom would end up based on the oceanic conditions in the water at the time. So you can see in the image up on the top left, image D and image H. So image D is the particle tracking model and image H is the satellite imagery, and these match up quite nicely. So we were fairly confident at that point that what we were seeing on satellite wasn't an anomaly, and that it was actually a phytoplankton bloom. So from there we went on to the inshore phytoplankton data from the monitoring team in the phytoplankton group in the Marine Institute, and we accessed their data, which is all publicly available. And we looked specifically in the southern stations because the bloom was obviously off the coast of Cork. And we could see that a harmful species called Karenia mikimotoi, or Karenia species in general, was at very low concentrations in the cell counts early June. And as the month of June progressed, the cell count started to increase. So it was at a few thousand cells per litre in the middle of June, and this jumped to 2.5 million cells per litre at the very, very end of June. So this coincided with, from our imagery, when the bloom peaked offshore and looked like it moved in towards the coast and moved around the coast of Ireland, showing up in Oyster Haven and Kinsale as well. Now, we couldn't specifically say that this was a Karenia species bloom at that point, but we were lucky enough then to have access to data from offshore, data from an NUIG phytoplankton cruise, that they actually went through the bloom as well, just at the beginning of July. And again, their data showed that Karenia species was the predominant phytoplankton observed in their samples. So we were fairly confident at that point that the satellite imagery was working and that we were able to validate it and say that that bloom that we've seen was actually a harmful algal bloom. So through the MFAF project, we've actually made this data now publicly available. And there's a link on the screen here, but I'll actually add it to the chat after this talk, where anyone can go on and have a look at the daily data that's uploaded there. And then every week there's a weekly composite automatically uploaded. So if anyone's interested in looking at those, feel free to go check out the link. And then just to briefly mention our satellite validation work that we do at the Marine Institute. So we have five hyperspectral radiometers mounted on our Celtic Explorer. And ultimately what these do is they take a remote sensing reflectance value combined together, And basically, this is the same kind of measurement that a satellite on an ocean or an ocean color sensor on a satellite takes. Obviously, the big difference being the distance between the satellite and the ocean surface. So the data from these sensors can be used for satellite validation, algorithm development. If we go through an area with a bloom, you can get spectral signatures from the different phytoplankton in the bloom, and water quality monitoring. But in the last few months through the MFAF project, we have spent a lot of time and research into improving our system. So we're collecting the best kind of quality of data that we can possibly. So we have, as I mentioned, sensors on the Celtic Explorer, but we put out a tender earlier in the year, and we have now procured new sensors for the RV Tom Crane. So in 2025, there'll be sensors on both vessels. We've also bought a robot frame in Plymouth Marine Laboratory that's designed specifically for this type of sensors. The sensors are attached to it, and it basically moves based on the sun's orientation. So this will minimize sun glint and ship shadow on the measurements, so it'll optimize measurement conditions. All of our sensors have been sent to Estonia to get fully characterized, which is basically a comprehensive evaluation of the instrument's performance. And finally, then we're going to use software that has been developed by NASA called HyperCP, NASA and EUMETSAT in Europe. They developed this as a community processing system. So having all our new kind of setup with the robot, the full characterization, and the new NASA software, we will have reduced our uncertainties in our data collection, improved the quality of data that's been collected, and in turn then our data will be available for satellite validation, which is a really good thing for Ireland, I suppose, to be able to put forward satellite validation data. And then briefly, just in the last minute, we have a new PhD student just started with us on the remote sensing team. This is funded through MFAF as well. She'll be working on phytoplankton functional types using remote sensing and hyperspectral data from our research vessels. And up to this point, the satellites that have been in space are multispectral. So I'll finish off with this video and explain over the video. Multispectral basically means we can only see specific colors in the water, but NASA have released or launched a new satellite called PACE, and this is a hyperspectral satellite, and this means they'll be able to see all the different colors in the ocean surface. And from that, we will be able to look at different phytoplankton functional types and differentiate between different species of phytoplankton at the ocean surface. So this is quite an exciting project that's just started, and hopefully in 2025, with our new hyperspectral radiometry system set up, we'll be able to produce nice products and satellite validation data, and hopefully have more products. So thank you very much. I'll leave you with that, with PACE orbiting the Earth there, creating a nice little video. I hope everybody could see the video as it's playing. So thank you very much. And if you have any questions, feel free to ask now or email me after.
Okay, one quick question, Catherine, from Stephanie online. Can you identify all species of plankton visible in the red spectrum?
Sorry, I'm just going to stop sharing there. So using the red band difference algorithm, we're looking at chlorophyll fluorescence. So we're not actually identifying different species specifically using that algorithm, but we are looking at chlorophyll. That's where the validation work comes in, and that's where you have to go to the in situ data and look at the phytoplankton data and see what's happening in the water at the time. Obviously now then with the release of PACE, NASA's new satellite, there are high hopes that we'll be able to differentiate between different species in the future. Their products haven't been released yet, but I think 2025 is when we'll see some phytoplankton functional products start to come through. But at the moment, the red band difference algorithm is a chlorophyll fluorescence algorithm. So we're looking at high fluorescing chlorophyll at the ocean surface rather than specific species. but we were able to differentiate to identify the bloom I talked about as a Carina bloom using in situ data.
Thanks very much. And Catherine, you'll be putting some links to where product can be found on our website in the chat, I presume.
Yeah, I'm going to do that now.
No, I think that's really useful because certainly on the south and southwest coasts, predicting potential blooms has been a thing for the aquaculture industry for decades. Right back in the 80s, trying to plan harvesting or movement of fish cages or whatever. So I think this is a really nice product that's now available to the industry for their support. Super. I'm going to close this talk now. Thank you so much, Catherine, for that.
Very much.
We're going to now move to the data collection framework. Linda O'Hay is the section manager in the Marine Institute responsible for the implementation and operation of the Ireland's data collection programme for fisheries data. Linda is also the national DCF correspondent for Ireland and is the contact point for exchange of information between the EC and Ireland regarding the preparation and implementation of our national work plans, which are annual work plans. Linda is unavailable to actually give the presentation, but she pre-recorded. So I'll ask Julie to hit the go button on this one. Thank you very much.
Hi, my name is Linda O'Hay. I'm a section manager within the Fisheries Ecosystem Advisory Services Group within the Marine Institute, and I am the programme coordinator for the data collection framework. And today I'm just going to give you a brief introduction into what the data collection framework involves. The data collection scheme comes under Priority 1, Fostering Sustainable Fisheries and Restoration and Conservation of Aquatic Biological Resources, under the EMFAF programme. The specific objective related to this work is 1.4, Fostering Efficient Fisheries Control and Enforcement, as well as reliable data for knowledge-based decision making. And the main aim or the type of action involved is to ensure compliance with Council Regulation 2017-1004, which is the DCF regulation, and Commission Delegated Decision 2021-1167, which is the EU MAP legislation. So the data collection programme involves collection and management of biological, environmental, technical and socio-economic data in fisheries and aquaculture sector. The Marine Institute is tasked with the implementation and the overall coordination of this data collection framework to support scientific advice required for the Common Fisheries Policy. This work involves cross-agency collaboration with both BIM, who are responsible for the socio-economic data collection in fisheries and aquaculture sector, and Inland Fisheries Ireland, who are responsible for part of the collection of the Adams species and also the Marine Recreational Fisheries Programme. This data collection scheme is a continuation of data collection activities that were carried out during 2014 to 2020, which was the EMFAF funded portion. The DCF itself has been in place since the year 2000 in various forms. But this new MFAF funded DCF takes into account additional components, which are new legal obligations and also the needs identified from the SWOT analysis and intervention logic carried out during the development of this new programme, reviewing the previous programme and looking at needs required for updates. So the needs identified through the SWOT analysis included fit-for-purpose data collection programme in line with new legislation, collection of transversal and spatial distribution of fishing effort for vessels under 12, including enhanced data collection on socioeconomic data for vessels under 12 metres, looking at specific knowledge gaps for data per stocks, addressing emerging stock assessment needs to improve advice. This includes management strategy evaluation, mixed fisheries analysis, ecosystem modelling, also looking at improving our sampling efficiencies and data management, and addressing specific scientific questions, looking at stock ID, mixing, migration patterns. The revised EU MAP legislation includes new regular activities, including recreational fisheries, looking at sensitive species, vulnerable marine ecosystems, social variables, stomach sampling, and also looks at end user needs agreed at a regional level, with an aim to establishing regional work plans coordinated by member states and regional coordination groups. The Data Collection Framework Regulation requires member states to collect biological, technical, environmental, socioeconomic data, as mentioned previously. And this robust scientific data is required to evaluate the state of fish stocks, profitability of different segments of the sector, and the effect of fisheries and aquaculture on the ecosystem. The Data Collection Framework Regulation sets out basic principles and general rules on the collection, management and use of data in line with the Common Fisheries Policy. It contains provisions for the multi-annual union programme and its implementation by member states, which involves drafting work plans each year. It includes communication between the Commission and member states through the national correspondence. I myself are the national correspondent for Ireland and the link between Ireland and the European Commission in relation to everything data collection framework related. It also outlines the role of regional coordination groups for cross-collaboration between member states, the storage and the sharing of data, and the support to scientific advice. Ireland's National Work Plan for 2022 to 2027 includes areas covering all aspects of the data collection framework programme. So biological data collection, which includes newly added at-sea sampling, which was initially driven by the COVID pandemic and proved a valuable data collection portal, and we now have a dual sampling stream where skippers provide data on catches. We've developed digital data collection for port sampling on commercial fish species in order to improve the quality of the data collected, and it's also trying to switch to paperless data collection. We're looking at optimising and maximising research survey outputs and continuing environmental and ecosystem data collection on surveys, increased modelling, simulation capacity for improved data limited assessments, and carrying out management strategy evaluations on stocks with limited historical biological information. It also now includes routine data collection on recreational fisheries, which is based on previous pilot studies carried out by IFI and now included in the routine programme. We also look at fishing activity data, impacts on fisheries, and economic and social data in both fisheries and aquaculture. And all of this data collection ensures good science supporting sustainability and providing sound evidence for policy. The DCF programme cycle and the various meetings that take place throughout the year all ensure that the data collection framework programme is on track and is assessed by the Commission and also by the Scientific, Technical and Economic Committee for Fisheries in 2 dedicated annual meetings per year. So work plans, which involves all of the data collection mentioned previously, are submitted to the Commission for review in October, and on the basis of STCF opinion and DG Mare, the Commission, work plans are approved by a Commission decision. Each year we're then required to report annually on the data collected, and these reports are submitted to the Commission by 31st of May deadline each year. We participate in various meetings throughout the year as well. The regional coordination groups meet annually. Within those regional coordination groups, there are intersessional subgroups that carry out work sharing information. National correspondents meet on an annual basis also to discuss work involved in data collection framework, and the regional coordination groups meet for their decision meetings and liaison meetings in Brussels in September. As I mentioned, there are regional coordination groups for the various areas, including North Atlantic, North Sea and Eastern Arctic. There's a regional coordination group on large pelagics, and for our case it's looking at tuna, and also a regional coordination group on economic issues. And this year, regional work plans for each of those regional coordination groups have been drafted and submitted to the Commission, and they're now incorporated into national work plans. So where relevant within the various sections of the national work plans, we add links to these regional work plans. So that's a whistle-stop tour of the Data Collection Framework Programme. For any further information and links, I've included the DCF website, the European Commission website, and also information on the regional coordination groups. Thank you.
Okay, that's super. I don't think we've any questions on that, so I'm going to move along. Our final speaker is Macdara Ó Cuaig. Macdara is a Connemara man raised by the sea, and he's been working in the Marine Institute for over 20 years. His main functions are in relation to collection of data in conjunction with Irish fishermen to help assess stocks for a sustainable future for the next generation of fish and fishers. So Macdara is working primarily within the data collection framework system that Linda's just described. So I'll hand over to Macdara for a talk, Sampling at Sea from Deck to Desk and Back. Thank you, Macdara.
Russell, and this is just a little bit on the end of that as well, ... infinitum, because it's goes on and on. It always there's a loop involved. So as Russell was saying, I'm involved in the data collection at sea. I'll be focusing on sampling at sea on commercial vessels from deck to desk and back. We will have another talk in the future on sampling at sea on surveys, MI surveys. So first of all, to kind of set the scene, what do we do? What do I mean by collecting at sea? Well, we work in the blue zone here on this slide. If you look over to the left-hand side, where the data that's used in the stock assessment cycle, there's the fishing data, logbook data, as Ali mentioned earlier on, port sampling, where we sample fish that come ashore before they go to market, observer trips, which is the sampling at sea, which is what I will be mainly zoning on here today, and then there's the scientific sampling as well on scientific surveys, which also give us trends that are used in the stock assessment process. So it's in the blue zone we're working. So what do we do? Well, we go to sea on fishing boats. These are pictures actually I took myself on a sampling at sea trip, where we work in conjunction with fishermen. We asked, first of all, my job is to ring fishermen, skippers and owners and ask them, will you take a sampler aboard with you, or will you sample at sea for us? And in this instance, this vessel agreed to take me out to sea, and you can see my clipboard there on the stack of boxes on the left-hand side. And what we do when we're at sea is we measure the catch. When we as scientists speak about the catch, we don't mean only the catch that is landed, we mean the whole catch, stuff that is not landed as well, for various reasons. It might be non-marketable, it may be undersized. There's different reasons why it wouldn't go to market. So when we go to sea, we measure the whole catch, and we go on a range of vessels. This was the day before I went on that trip. This is the boat I was on here, a Greencastle boat, Northern Kelt, there on the right-hand side in the blue. So that's a large demersal trawler, which we would do many trips on demersal trawlers within our fleet. Then to the left of her, you'll see a small inshore vessel that actually targets herring. We had a sampler at sea on a herring trip actually yesterday. Then there's a larger, the black and red vessel there is a polyvalent pelagic vessel, which fishes both demersal and pelagic fisheries. And the blue and black one on the left there is a deep fish, well, further offshore fishing crab vessel. So that shows you just in that one photo alone the range of vessels that we go to sea on and that we sample data. So when we go to sea, what do we do? Well, first of all, there's two types of data that we collect from the deck. We have sampler at sea data, which is where we have a trained sampler on board with the fishermen during their trip, where we're working alongside the skipper and the crew, staying out of their way so they can do their work as efficiently as possible. As Ali mentioned earlier on, we don't want to be obtrusive. Even when we're present at sea on the vessels, we try to be to be staying out of the way of their natural workflow, while at the same time completing our work as efficiently as we can. And we also have at-sea self-sampling, which is where skippers and crew collect data for us, and they bring samples and data ashore. This was kind of developed or to the stage it's at now, really during COVID, where we couldn't send persons to sea for health and safety reasons. So the fishermen, to help us plug a gap that was emerging from our data set, we worked together and we set up the at-sea self-sampling program for offshore fishing. And that works well, but it's by working together that we bring the data and the samples ashore. Obviously, when the samples come ashore, then our teams collect them off the boats and we measure them and work them up, take atlas, all that kind of stuff. So the types of fisheries that we're involved with: pelagic fisheries, which are mackerel, herring, boarfish, sprat, that kind of fisheries. Demersal fish like cod, haddock, whiting, monkfish, and inshore or shellfish fisheries as well, like Ali mentioned earlier on, razorfish, crab fishing, lobster fishing, crayfish, that kind of stuff. And then what's interesting about this is that what we need to do is we need to increase our coverage, and our coverage has been increasing over the last number of years. Sometimes it can be difficult to get trips in certain areas or métiers. For example, you may have limiting factors such as space on a vessel, where we couldn't get a sampler on a vessel because we wouldn't have space, but that's where our at-sea self-sampling comes in. But then some métiers, they're not, our current system wouldn't allow for comprehensive enough at-sea self-sampling for the data that we need, so we need to have people aboard. So we have to manage it with the fleet and with the crews. And I'd be calling skippers, and it may be that this month wouldn't suit, but next month might be fine for a trip with them. So as I say, we are on a range of vessels on a range of different species. This is just a kind of a montage that I put together from trips that I was on with people, inshore and offshore. Because, as I said, not all the data that we require comes ashore. So when we go to sea or when we get samples from the skippers, what do? We collect data on age, composition of the fish population, the number of fish that are removed. We can see that from the unwanted catch as well, and the wanted catch. And also we get the age composition of the unwanted catch. There's wanted catch and unwanted catch. And we need to get all of them to get the full range for our data assessment. So that's for the green zone where the data is collected. Now, as I said earlier on, we do this in cooperation with the fishermen. We don't do it on our own. We couldn't do it on our own. And even when we have the skippers and crew self-sampling for us, we're working in conjunction with them. We actually use WhatsApp as a kind of a live QC, working with the skippers, where they record their data, take a photo of the data sheets, they send it on to us. We do a quick quality check and give them live feedback. A few years ago, I was at a conference quite far away, and I was still talking to fishermen on the Porcupine Bank via WhatsApp, working with this kind of system, which ensures then that the data is quality assured in situ, even though you're hundreds of miles away. And what that does, it ensures that we don't have the same error propagating through all the trip. We nip it in the bud at the start of the trip in conjunction with the skipper. And we do the same with our samplers as well, especially if we have new samplers. They use WhatsApp and different technologies to get onto us, just to verify that they're collecting the data to the quality and standard that we require. So then once we've collected the data, then what's the next step, you'll ask? Well, then we go into this amber zone here, which is working with colleagues as well, international colleagues, where the data sets from Ireland and different nations are brought to ICES for the stock assessment process, because it's not just Irish vessels that fish off the west coast or the southwest coast or the north coast. So we bring all the data together at the working groups and it's collated. And then from that, then we move into the yellow zone. So the yellow zone is up here, up above, and the yellow zone is where the data is given to the managers, and the politics comes into it as well. And you may have heard yesterday there was an agreement between the EU and UK on opportunities for next year. So our assessments process, working with our international colleagues, gives us the size of the cake, and then the slice of the cake is what the quota that's given to each nation, and then back to the boats again, where they're sliced, that they can go to fish next year, and provides extra sampling opportunities for us, and off we go again. So what's next with this DCF program? Well, we want to increase participation, increase the number of trips. We did analysis a few years ago, and it's better for us to have more short trips than longer trips, because it's better data if you go on different types of vessels, because it spreads the effort across the fleet. We want to continue awareness, which our self-sampling program is helping with this, because it makes the skippers who are involved and very aware of the kind of work that the scientists do when they're on the deck as well. And actually, what we do find is that we might have a scientist sampler at sea on one trip, and then the next trip, then maybe in the next month or the month after, the skipper would do a self-sampling trip for us and would have learned from when the sampler was aboard. As I mentioned earlier on, we collect quality data from the deck, and it's very important. And even when we have questions, we go back to the deck, and the shorter the time span between the data being collected and it being checked, the better, which is why we use our WhatsApp quality check. So for the future, it's more trips. And for the future, this image here I put up because I think it encompasses what we do. This is the sun setting in Galway Bay in the west, and you see this young lad, my young lad, actually running towards the east, running by the sea. So that's our infinity loop here. We're not doing it just for ourselves. We're doing it for the sea, for the fish, for the future, and for the people for the future. Gurmila Magi. I'll just keep it nice and short.
Super. Thanks very much, Macdara. I have a question from the chat for you before you go for your lunch. Do the vessels have a choice to do self-sampling or to have a sampler on board? Can they do one or the other, or how do you work that?
Yeah, they can do it. The vessels have a choice to be involved or not to be involved. In other nations, there's a compulsion on vessels to be involved with data acquisition. Now, under their license, they're encouraged, obviously, to be involved in the scientific process, and it's an investment in their future to be involved. But they have a choice between self-sampling or sampler on board. We may find that some vessels, this trip, it suits them to have a sampler on board, or it may not, because sometimes if they're prosecuting a certain fishery, they might carry more crew, so they wouldn't have space. So they do have a choice, yes.
I think that's it. Okay, so hopefully we will have the recording available in the next few days, and I'm sure Julie will send that on to everyone who registered on the talk. At one stage, we had over 124 people online, which is a record for us. So thank you very much. Can I just thank the support people for setting the whole thing up? And can I also thank the speakers again? Very interesting talks. And we plan to have one next year, two next year, probably April, May time, and then again in November, December. Okay, I'll close that. Thank you everyone for attending. Thanks to the